All right. Looks like we're right at the top of the hour, so we're going to go ahead and get started. First of all, hello everyone, and welcome to the webinar. And thank you so much for joining us today. We're really excited to have you here. My name is Kayla Ripple, and I'm a senior associate with the Lenfest Ocean Program. So for those of you who may not know us, the Lenfest Ocean Program is a grant-making program. We fund research projects and expert working groups to address priority science needs that are facing ocean and coastal decision makers. So to learn more, you can visit us at lenfestocean.org. There's also a short video on there about how we work and the kinds of projects we support. So definitely check that out if you do visit our website. And while you're there, you can also sign up for our monthly newsletter, which contains updates on new and continuing projects. And if you're on social media, I'd also encourage you to follow us on Facebook and Twitter at Lenfest Ocean. In fact, we'll be live tweeting this event today. So if you'd like to, you can follow along or interact with us there using the hashtag LOPwebinar. For today's webinar, we're very excited to have joining us Dr. Stephanie Green, Natasha Hardy, and Miram Gleiber, all with the University of Alberta, and Desiree Tomasi with the NOAA Southwest Fisheries Science Center. Larry Crowder is also a project PI, and he's joining us for the Q&A section or portion of the webinar. So we're very excited that he's here with us as well. Now I'll let the researchers get into the details, but for us at Lenfest, this is an important project that is helping to inform how fisheries management can address the issue of shifting marine species. As ocean temperatures warm, this becomes an increasingly apparent issue, pushing marine species to new habitats and altering population distributions. Over the past 3 years, this research team and their collaborators have been developing modeling methods that account for predator-prey interactions to inform projections of where predators may shift due to climate change. And they're using albacore tuna and their prey species as a case study. My colleague Emily Knight will be dropping a link to their project fact sheet in the chat, as well as a link to their latest manuscript on using trait-based approaches in global change ecology. So we're really eager to hear from them and for them to share some of their preliminary results, and also a new open access trait database that's available for others to use now. We think these products will be beneficial in helping to inform future management strategies for albacore along the Pacific coasts of the US and into Canada. As this project is not yet complete, we'd encourage you to reach out to us at Lenfest or the research team with any thoughts, feedback, or questions that you might think of after the webinar. And just before I turn it over to the panelists, I want to go over a few webinar logistics. First, all attendees are muted to prevent any feedback or echoes during the webinar, but we do want to hear from you and are saving time at the end for questions. So to submit a question, you can type it into the Q&A panel at any point during the webinar. I'll be keeping track of these and we'll read them aloud at the end for the panelists to answer. If you have something other than a question, like a comment, a statement, or a resource you'd like to share with the speakers or the attendees here, you can type that into the chat box. We'll be sharing links to other project-related materials as well in that chat, so keep an eye out for those. And finally, this event is being recorded. I'll distribute a link to the recording next week, along with links to all the materials that we share in the chat. Once you get that, please feel free to share this with anyone in your networks who may be interested. And with that, I'll turn it over to Dr. Stephanie Green to get us started. Take it away, Steph.
Thank you very much, Kayla, for the introduction. And thank you everyone for being with us this afternoon. We're very excited to share some updates as well as the rationale and some early findings from a project we've been working on. We're now in our 4th year of this work funded by Lenfest with a number of wonderful partners. And the focus is on the potential for traits to help provide a useful lens in understanding how ocean species and ultimately those that support fisheries will be changing in our oceans as climate change intensifies. I want to first start by acknowledging the many partners who have contributed in many ways to this project, both at the University of Alberta, at Stanford, at the University of California, San Diego, and our many partners at the NOAA Northwest and Southwest Fisheries Science Centers. Some of these team members have been with us since the proposal phase, and others have joined along the way, contributing data, ideas, analyses, and context to the projects that we're working on. And you'll hear more about many of these team members as we walk you through what we've been up to and some of those initial results. All of us are motivated as scientists to want to generate information and tools that help us understand how ocean species are changing as our climate changes. This is a study that really piqued my interest and impressed upon me the urgency of these questions that came out a number of years ago now, looking at how our wholesale levels of diversity may shift in the ocean as a result of future climate change. These are projections for changes in species richness, the number of species found in a particular place under RCP scenario 8.5. And as you'll see in the cool colors, some places are forecast to lose species, to have declines in diversity, and other areas are forecast to have increases in the number of species there as organisms move in as their thermal habitat shifts. Well, these wholesale changes are likely to have large implications for coastal communities and for the resources that we rely on that come from the ocean. In particular, subsequent work that's focused on fisheries that target many species in ocean systems have revealed that we're likely to see many of the predators and species that support fisheries shift across international boundaries as a result of the changing climate that we see in the ocean. And these changes are not necessarily far off into the future. In the coming decades, we'll see that management and that various groups that rely on these resources are going to have to grapple with this idea of changing species distribution and what it means for the abundance and location of species that might be targeted. And so as scientists, we're really interested in understanding how we can provide information and tools to understand when and where these shifts will occur. Typical approaches that we use to understand species redistribution in the ocean use information on past environmental conditions, often abiotic conditions like temperature, dissolved oxygen, and other parameters, and relate that information to both the distribution and movement of species in the ocean. And then to look ahead, we would use the relationships between species distribution and movement to infer how species might shift under projections of those same variables. Increasingly powerful modeling tools allow us to account not only for changing abiotic environmental conditions, but also the many interactions between species that might moderate these effects, in particular interactions between predators and prey. And so we're seeing ecosystem models become increasingly powerful tools to try and understand how both a changing ocean climate, as well as shifts in the distribution of resources like prey, might influence top predators that are contributing to fisheries around the globe. One of those modeling frameworks that's being increasingly used is called the Atlantis ecosystem model. It's what we call an end-to-end model that considers dynamics in the ocean in terms of climate and oceanography, how those dynamics propagate up through the food web, influencing the distribution and abundance of species and their interactions with one another, and then ultimately what those dynamics mean for the fisheries that many species within the food web support. And so with a large model like this, scientists can conduct scenario analyses where you might investigate the effect of both changes in the fishery or policies that affect a fishery, as well as changes in ocean climate state and how that might play out in terms of the abundance and distribution of species in the particular ocean space. Important connections within a model like this are those between predators and prey. So highlighted here in the red box, a single arrow representing the relationship between one prey species or group of species and a predator that might consume that. And these relationships are incredibly important to the internal workings of the model, and also something that model output are quite sensitive to. In order to parameterize these relationships between predators and prey, scientists often look to diet analysis of predators to see what they're eating and how that changes in space and time. This is often done through gut content analysis, a type of work that myself and colleagues have done in many different systems for a number of years, and it provides vital information on the relationships between predators and prey. Of course, it's not without its challenges. In particular, by nature, this type of work is retrospective, giving us an idea of what a particular predator is consuming in a time or place. It can be often resource and labor intensive to do, collecting both the samples and IDing potential prey items to a fine taxonomic resolution, ideally to species in our ideal world. And what we're doing with these data is characterizing prey use by a predator, but we don't necessarily have a picture of how what a predator is selecting to eat might relate to what might be available out there in the environment, and therefore how that diet might shift as the prey field available to a given predator shifts. And so as we're looking at increasingly novel combinations of species interacting with one another, as scientists, we're wondering, how can we provide additional insights to information that's collected through processes like diet analysis work to understand who is going to eat who in a changing ocean? So what our team has stepped back and done is thought about the way that foragers really consider the potential prey they have in front of them. So I want to give you an example of what is a daily choice that I face here on campus at the University of Alberta. What am I going to snack on? I might look at a couple of different options that I have for my mid-afternoon snack, and they might vary in a number of aspects, which we might call traits, both in texture and color and nutritional profile and in cost. And of course, myself being a pretzel girl, I'm going to consistently choose something that's in line both with my budget and also in terms of the traits that I tend to prefer. If we were to observe my snacking habits over time and across space in relation to what I might have available, we could use that information to think about what someone like me might choose if I was put in a completely different context, perhaps going to a grocery store in a country I've never been to where the same options aren't available. So these kind of trait-based analyses, where we're using information on the characteristics of organisms to infer how the impacts of global change will play out, are increasingly being done in ecosystems across the world. In fact, in a recent synthesis that we did, we found an exponential increase in the number of trait-based papers that are being published looking at the effects of global change across the world's ecosystem. This is a technique that really originated and is still largely used in terrestrial ecosystems, largely by plant ecologists using traits of plants to describe where organisms will be found and who they'll interact with under future climate states. But we also believe that this trait-based lens can provide really useful information and understanding in ocean spaces how predators and prey will interact in the future. And here's why. Let's look at the predator predation process that happens for all predators that are out there searching for food. That predator must encounter different prey items, then attack them, capture them, and ultimately consume them. And if we're thinking about predators in ocean spaces, in particular our gape-limited visual predators, which comprise most of the fish species that we have in the ocean, we can make predictions about traits of particular kinds of prey that might influence this process. For example, the habitats that those types of prey might occupy in terms of water column position, their activity patterns might influence encounter rates with a given predator. We might predict that physical and chemical defenses, coloration patterns, or the extent to which prey items might aggregate together could influence the extent to which a predator attacks those prey. And we might predict that the size, shape, and nutritional profile of different prey items might influence the extent to which they're captured and ultimately consumed by a predator. And of course, these example traits aren't mutually exclusive between steps, but this is the framework that we're working in to generate some hypotheses that we can test. The idea of using traits to understand generalities in predator-prey relationships has been demonstrated in other systems recently. In particular, I've been involved for a number of years in work with the invasion of Indo-Pacific lionfish, which are spreading throughout the Western Atlantic basin and interacting with diverse communities of marine species as their range expands. One of the challenges with lionfish is that they're a gape-limited predator that consumes a wide array of species. And so as we think about the impacts of the invasion, we need a way to identify which species are likely to be most vulnerable and where impacts will be most intense. Applying a trait-based lens to this problem has allowed us to identify that species that are solitary, slender, small, and nocturnal are orders of magnitude more likely to be consumed than species without these traits. And they're trait sets that are shared by a relatively small number of taxa across a few families relative to the huge diversity of species that lionfish encounter in the system. Applying this trait-based lens has allowed scientists to look ahead of the invasion front to areas where lionfish have not yet expanded into, to identify places that have taxa with these set of vulnerable traits, and to suggest those areas as priorities for monitoring and for management intervention. And so this trait-based lens has allowed us to make predictions about where interactions are going to be strong, and then integrate those predictions into suggested strategies for managing the strength of those effects. So of course, biological invasions are not the only way that predators are expanding their range. As we've discussed, climate change is intensifying in our oceans, and we're already predicting that species are going to be shifting. This is a study that projected the distribution of several hundred species in the Pacific Ocean, forage species in the system. And here the radar plots, the extent of the line and the direction, give the direction and magnitude of the predicted shift. As you can see, species are likely to shift, but the direction and magnitude is not consistent. And so taking our predictions to this scale suggests that we also are going to see new combinations of species encountering one another. And what we're wondering is, applying this trait-based lens to the system, can we come up with some ways to understand how predators that are foraging in this dynamic system might utilize prey in the future, and what the consequences of that prey utilization might be for abundance and distribution? One of the species that we're particularly focused on in terms of understanding the consequences of changing forage availability is albacore tuna, a commercially valuable species distributed in all oceans across the world, but for which we have evidence that there perhaps is already shifts in their range being seen. So these are data that give the catch per unit effort of albacore in the Eastern Pacific fishery. What we can see is that essentially the center of that fishery has been shifting northward, and then both offshore as well as inshore, perhaps in relation to changes in climate states, but perhaps also in terms of changes in prey availability. And so in our project, what we're focused on is using albacore tuna as a model species to try and understand the extent to which traits can inform our understanding of how species abundance and ranges will shift. Albacore are highly migratory, they're commercially valuable, and most importantly for our project, they have a broad and variable diet that we suspect changes across environmental gradients, meaning that we might be able to use traits as a useful lens to distill that diversity into some general synthesis that we can use for future prediction. The goal of our project has been to develop and test a framework for integrating trait-based insights, and albacore tuna and the California current are our test case. Our research has been unfolding in 3 phases. First, we've been characterizing the trait basis of albacore foraging dynamics through data synthesis, and then applying those trait-based insights to hindcast the trait basis of foraging dynamics in the California current system specifically. And ultimately, the goal of the project is to use those insights to inform models that forecast the distribution and abundance of albacore into the future. I'm going to turn it over now to my colleague, Dr. Natasha Hardy, to talk more about this first phase of work that we've been doing, the data synthesis phase.
So we identified a need for data on prey species traits and generated a large data collection and synthesis effort. You can go to the next slide. We first sought to quantify albacore predator-prey interactions, having actually no number on their prey interactions. So we formally investigated, what do albacore consume worldwide? We can go to the next slide. To do this, we reconstructed the diet composition for albacore, and we found 26 publications and reports dating back from the 1880s even to 2020 across the world's ocean basins, and containing 69 individual diet tables such as these, which included sometimes multiple metrics for diet composition and often required lengthy digitization and translation steps. So that enabled us to confirm that obviously we did know to a certain extent that albacore tuna have taxonomically broad diets. We've been able to put a number to this. In this phylogenetic graphic, we illustrate over 308 species recorded in albacore diets. And these interactions are typically simplified to model a subset of the most abundant species. We argue there is a need to model change, though, in complex ecological communities such as this, using generalizable functional traits in order to capture variability occurring in the broader prey assemblage. But for that, we needed to systematically collect trait data for over 300 species, and thankfully didn't do that alone. So Miram's going to talk to you about that in the next slide.
So in order to apply trait information to better understand relationships between pelagic predators like albacore and their prey, we assembled the Pelagic Species Trait Database. We released this open source last spring on Borealis, which is the Canadian Dataverse repository. You can scan the QR code with your phone to link to it right now, or see the link that's going to be in the chat. The database is global with 521 species of fishes, cephalopods, crustaceans, and other invertebrates. We included species consumed by albacore tuna globally from the work Natasha just presented. And these species are useful beyond just studying albacore, since they have broad diets that overlap with many other pelagic predators. Other species are pelagics more specifically from the California current system. I also want to highlight that the traits are life stage specific, so separate traits for adult, juveniles, and larval stages. We have over 40 traits that represent habitat, behavior, morphology, nutritional quality, and population status. I'm just going to briefly highlight some traits we've been using in our traits-based analyses as key influencers on parts of the predation process. These phylogenetic trees show the spread of trait coverage and trait forms across all the species in the database. Horizontal and vertical habitat position, as well as activity patterns like diel or seasonal migration, impact encounter rates between predators and prey, since they need to physically overlap in space and time. Morphological traits for coloration, like counter shading, silvering, and transparency, impacts a predator's ability to recognize and attack prey. We also collected morphometric ratios that are a continuous numerical way to examine body shape. These morphometric ratios have also been useful for species and stage-specific conversions from length values of different types, for example, standard length or mantle length to total length. And nutritional quality impacts many aspects of the predation process. We include lipid content, protein content, and energy density standardized to wet weight. Of all the traits, nutritional content is the least resolved, as you can see these gaps on the phylo tree represented by the gray spaces. For the majority of these, we actually can't find any values in the literature. And this is an area of the database we're focusing the most on filling in gaps and learning more about nutritional quality variation for important species. Before going any further, I'd like to acknowledge the students from our lab who truly made this database become the resource it is today. Traits were primarily collected from the literature based on keyword searches for each trait, species, and life stage. We also used other online resources like FishBase. We collected morphometric ratios by measuring multiple dimensions from images found online, confirmed to be the correct life stage and species. And recently, we've added in nutritional analysis because, unlike many other traits in the database, nutritional traits are also not static. And recent work from the California current is showing that forage species can vary in nutritional content across seasons, years, and likely many other variables. Our master's student, Alana, is currently exploring this variability in nutritional quality in key albacore prey. She's pictured here at Scripps this past summer, working in collaboration with Anella Choi and Elon Portner, running samples for energy density with bomb calorimetry. And this fall, she ran the same samples for lipid and protein content and is analyzing the patterns of variation within and across species. I also want to highlight that the database includes documentation on data collection materials to serve as a framework for the augmentation of this database in the future and to make it reproducible in other systems and for other predators. We're currently working on releasing an updated version, and along with that, we'll have an in-depth tutorial available online. We'd love to hear from you if you're interested, have questions, feedback, or even data to contribute. And now I'm going to hand things back over to Natasha to share some examples of how our team has used this database to study the feeding dynamics of albacore tuna.
Thanks, Miram. So using that prey trait information, here we sought to expand on descriptive diet analyses for albacore tuna, and this is for that global meta-analysis. And here we wanted to identify and test non-taxonomic predictors for that broad biodiversity in albacore diets. And I focused here on 4 of the habitat association traits, the seasonal and diel migration behavior, as well as horizontal and vertical habitat use or water column position, as they contribute to the likelihood of the predator encountering their prey. And these were used to synthesize that diversity to 7 different prey trait guilds. Beyond taxonomic diversity in diets, this allowed us to plot the historical diet composition of albacore here using a relativized metric of frequency of occurrence data, because that's the data we consistently had for the longest time series historically. And to illustrate the contribution of these trait guilds to the diets from these different locations sampled. Both trait guilds and individual trait information significantly explain variation in albacore diets across locations sampled, meaning that trait information presents us ecologically relevant and useful classification information for albacore prey that will likely provide us with indicator or explanatory information for diet shifts for albacore and other broad-ranging prey. For example, within this historical time series, we can see in the North Pacific a high contribution of coastal and shelf pelagic species, as well as mesopelagics, and similar patterns for different trait guilds are visible in other ocean basins. So we found this historical variability in trait-based diet composition intriguing. And we wanted to know what is explaining these shifts and patterns. But we can't go back in time and collect all the variables which we think could explain these shifts for all of these data. But we do have some of these data in the California current system, and being in Western North America, we're now going to focus on the North Pacific in applying trait-based frameworks for modeling predator-prey interactions. The California current large marine ecosystem is an eastern boundary current system characterized by seasonal upwelling, a strong oceanographic gradient, and with great data on historical climate oscillations. This is also where data from 3 modern albacore diet studies are available and provide an extension to those historical data. Earlier intensive studies by Sarah Glazer and Dan Madigan span 2005 to 2010. And these are augmented by an ongoing long-term sampling program led by Kat Nichols at the NOAA Southwest Fisheries Science Center spanning 2007 to present. Also available in this region of the world are a breadth of systematic scientific surveys, enabling estimation of forage community abundances. We use data sets from annual NOAA surveys that use midwater trawls towed at 60 meters depth, and they're sampling within a similar range of foraging habitat for albacore. For the Californian part of the system, we use data from the rockfish recruitment and ecosystem assessment surveys managed by Jared Santora and John Field. And for data offshore of Oregon and Washington, the Stock Assessment Improvement Program, as well as its replacement, the Coastwide Cooperative Pre-Recruit Survey, were made available by Rick Broder, Brian Wells, and Toby Orr. So we'll be using the California current system to move forward from our data synthesis phase and into our hindcast modelling phase to model albacore foraging dynamics in relation to a suite of environmental and explanatory variables. At first, we focused on coupling albacore diet composition data, forage composition, and trait data to investigate relationships between predator resource use and environmental abundances of those resources in the California current. Note that we are considering data from 3 subregions, and these are based on physical oceanographic differences in these graphs illustrated by the dashed line. We seek to understand what resources are shared by or differ between albacore and surveys of the system, and of course how prey traits can help us move forward beyond high species diversity in the system to understand what characteristics this predator may be encountering and even selecting for beyond what could be available to them in the system. In order to integrate 3 surveys and 3 diet data sets for comparative analysis, I just want to quickly highlight that we did obviously need to standardize taxonomic IDs to be referring to the same taxa, standardized counts to be comparing proportional abundance of taxa within diets to within surveys on a per sample basis. And we then aggregated rare taxa. We only use taxa in analyses that contributed to greater than 1% of diets or surveys in any region. So rare taxa, that means over 158 species were combined into 32 taxa, and that only affected 4% of the diet data and 6% of the survey data, meaning the majority of the data were already accounted for in the 30 plus pre-taxa. So that these bar plots display the mean numerical proportions of the 32 taxa observed in diet and survey samples, here as an average for each year and region. And from these data, you can see the interannual and regional variability in both data sets. There's evidence of prey switching in the diet data across years and variability in environmental forage abundances, such as that increase in gelatinous zooplankton in the orange or ochre tones visible in the survey data after the marine heatwave of 2014. That same signal is actually not visible in the diet data. So we can see that dietary proportions are different to the surveyed proportional abundances of forage. Another visible example is that of euphaustids in the dark green, which dominate the survey samples, and they are actually relatively common in albacore diets, especially in the north of the system, but to a lesser extent than in surveys. So in our multivariate community analysis of these data, each point here represents individual albacore stomach and trawl sample composition, given proportional abundance of taxa in each sample. The principal axis that explains variance in these data is that difference between diet and survey samples. To a lesser extent, we did observe differences between region and year with that different polygon size in the central California current system. And I'll note here that partly due to shifting fishing effort and sampling effort, and because albacore distribution is shifting northward, we are increasingly lacking diet data from the central and southern parts of the system. And the majority of our data do now come from the north of the system. So despite some regional and interannual variation, diet samples consistently occupied a broad swath of the top right of the ordination space, while survey samples were more central and to the left-hand side. So the main difference between these data is also in the size of the polygons or standard ellipse areas, whereby albacore diets do overlap with the surveys, but also sample more broadly than the systematic surveys do. Several key species were consistent in explaining that variance. So for diet samples, chub and jack mackerel, sorry, gonaded squids, boreal clubhook and armhook squids, anchovy and sardine were more abundant in the diets than in surveys. Overlapping taxa included myctophids, barracudinas, marked squid, hake, and rockfishes. That's still quite a number of species, and to model in previous efforts to model albacore distribution in relation to abundances of these taxa have struggled to identify significant relationships. So this leads us to ask of these data what non-taxonomic information explains patterns in these assemblages in order to identify potential ecological indicators for albacore diet composition. So we modeled the relationship between trait information here for habitat use, morphological and nutritional traits, directly to the explanatory variables, starting with region of the California current, year, and diet versus survey explanatory variables. As an overlay on species assemblage data, so we use a multi-matrix model known as a RLQ and fourth corner technique. And this model fits a penalized likelihood term that shrinks non-significant parameters to 0, and that's represented by white space. And significant interactions are then displayed in a heat map with positive correlations indicated by warm colors and negative correlations indicated by cool colors, and the strength of the color representing the strength of the modeled relationship between a trait value and an explanatory variable. So forage resource composition variance was significantly explained by all of these fitted variables. Displayed here are the results of the diet and survey signature in the data, which was the strongest fitted relationship and explanatory variable, followed by year and region. Here in blue are traits that were negatively correlated with diet and also characteristic of the survey data, and that includes oceanic and dull migrant taxa, as well as transparent colorations and physical defenses, so that the Faucet signal is visible there. And in red were traits that are positively correlated with diet data, especially coastal and mesopelagic taxa, counter shading and silvering coloration, and especially higher energy density values. Those relationships are tempered by interannual variability. As you can see, these colors are not as strong, and the relationships were not as strong as the relationship between diet versus survey differentiation. Lastly, for this analysis, relationships among regions, there were expected differences between these regions. The north column on the left is not significant because that was the most data-rich region. So the data you see in the year and diet columns are largely driven by the north, with coloration in the central and southern columns here indicating very minor differences to that overall signal that is visible from the north data set. So the main take-home message here is that several habitat use, morphological and nutritional composition traits significantly described albacore resource use in different proportions to those observed in systematic surveys of the forage. And both in these data and actually in that historical time series, there's evidence for relatively stable use of some of these traits, such as the bright red colors here, coastal mesopelagic taxa and of higher energy density value. We're going to go back to Miram to hear more.
So now we know that albacore and surveys are sampling differently, and the proportions of prey they consume are not necessarily reflective of the proportions in the environment as sampled by trawls. Next. Albacore are feeding more broadly, also including species in their diet that are not well sampled by trawls, likely due to them existing deeper or shallower in the water column. While albacore are considered generalist predators, they do appear to have some selective tendencies, since they are taxa abundant in surveys that are not consumed by albacore, and traits explain these preferences. We wanted to further explore these relationships with a single descriptive metric, so we used an electivity index to represent the magnitude of similarity or difference between the proportion of prey in the diets and survey. The index we used is Vanderploeg and Scavia's relativized electivity, which is recommended for use in natural environments. We evaluated this index of prey electivity, also called Estar, from both a taxa and traits-based perspective. Estar ranges from -1 to +1, with 0 being neutral. I'll be using warmer colors for positive indices, indicating higher relative proportions in the diet, cooler colors, negative indices, thus lower proportions in the diet. Colored circles show instances where electivity could not be calculated, since the taxa or trait is either absent in that given year in the diet, indicated by blue circles, or in the survey, indicated by red circles. And based on the outcomes of a community analysis, we did exclude some species that were poorly sampled by the surveys. But that actually only excluded a small portion of the data, which allowed us to examine the relative prey electivity, considering almost anything a tuna might encounter in the water column. For the trait-based approach, we included 13 traits known to impact the predation process, and the same traits in the RLQ fourth corner analyses you saw earlier from Natasha. This plot is showing the taxa-based results in the Northern California current, where we have the most years for comparison. Taxa are listed highest to lowest based on overall mean electivity values across years, and there's a lot of annual variability. However, we do see some taxa were more consistently consumed at higher or lower proportions compared to surveys, and some are highly variable. And this tells us something different from considering diet information alone. For comparison, I'm now listing on the taxa their rank in terms of mean abundance in the diet in this region. The highest positive index values were for sardines, the clubhook squid, and octopuses. While these taxa are not as abundant in the diet, we now show they are indicators of a high interaction strength with albacore, with some evidence for selection. In contrast, many taxa that are more highly abundant in the diet had variable electivity or consistent strong negative index values. And for the sake of time, I'll just be showing results in this region, but know that in the South, we see similar patterns, just represented by different taxa. But tuna don't see taxa, they see traits. So let's put on our trait-based glasses and see what prey electivity looks like from a tuna's perspective. This heat map is showing trait-based electivity in the Northern California current. There's a lot going on here, but don't focus on that. Instead, I want you to unfocus your eyes as if you're looking at a Where's Waldo. What we see is a lot of consistent colors across rows, indicating consistent electivity patterns for traits across years. In fact, 9 of the 13 traits have trait forms that are consistently highly selected for by Albacore, with the alternative trait form having high negative selection. And just for fun, let's take a peek at the South. The pattern is actually the same as the North. The same trait forms were positively selected across years. And these are the same trait outcomes from the RLQ fourth corner work as well. Next. These density plots are showing the relative amounts of variance in Estar, so electivity, across years from all taxa compared to all traits in each region. And we see that the interannual and regional variability in albacore prey electivity is ultimately minimized through a trait-based perspective. So after combining multiple large data sets, we see that in this variable diverse system, for a predator with variable diverse prey, the same traits are selected across years and regions with vastly different environmental states from 2 different analytical approaches. Now that we've given an update on our results, I want to zoom back out to the broader project. What we've been focusing on for the last few years are these traits-based approaches, from data synthesis, including the creation of a trait database for prey of pelagic predators, and a trait-based examination of global albacore diets. Then hindcast work using traits to look at the last 2 decades of albacore feeding patterns relative to prey sampled in the system. And now I hope we've shown you today that across the highly variable complex diets of albacore tuna, traits not only explain feeding patterns, but actually simplify spatial and temporal differences. So now that we can use traits-based approaches to describe albacore predator-prey interaction patterns in the California current, we can work toward integrating this information into forecasting changes in albacore distributions into the future. A little peek into the next steps, where we are modeling albacore diet shifts across the variable environmental conditions of the California current. Pivoting off the work we presented today, we're using a set of taxa and traits that are indicators of a high interaction strength for albacore to examine if environmental variables mediate the role taxa or traits play in explaining diet composition. And fortunately, our collaborators from Future Seas Consortium are actively working on past and future projections of climatic change in this region and the subsequent changes for important prey species. Ultimately, we're interested in seeing if similar or different traits are preferred by albacore across environmental regimes. One option is there's no relationship, but our analyses so far provide evidence of some consistent preference by albacore for traits over time. And if traits are conserved across climate states, then albacore feeding can be highly predictable, or they may switch prey with climate states, and traits can also provide predictive power in distilling these relationships. Then using these hindcast trait relationships will allow us to look forward to make future predictions about how albacore populations will be impacted by changing environmental conditions. And to speak more about how this work fits into the broader efforts by NOAA to study changes in the California current is a lead investigator from the NOAA Future Seas Consortium, Dr. Desiree Tomasi. Future Seas is a collaborative interdisciplinary effort to explore potential impacts of climate change on West Coast fisheries and to evaluate strategies for managing those impacts.
Thank you, Miram, for the introduction, and to the entire project team for inviting me to participate in this presentation. I will provide a brief overview of our project, Fisheries, which, as Miram said, is an existing NOAA-funded project that is developing climate and ecosystem projections to facilitate climate adaptation in fisheries management. The current phase of our project is focused on the forage complex and specifically on anchovy, sardine, and market squid. The title of this phase is Impact of Climate and Ecosystem Change on the California Current Forage Complex and the Fishing Communities and Predators it Sustains. Next. We know that the climate will be changing, and the Fisheries team has actually developed downscale projection of ocean conditions for the California current. So we've taken 3 Earth system models on the left and 1 RCP 8.5 scenarios, and integrated them with a ROMS model, which is from the University of California, Santa Cruz, to generate projection of what the oceanography in the California current will look like up to 2100. And if you want to know more about that process and that methodology, I encourage you to read the paper there by Merposa-Buil et al. In 2021. But what is apparent from that work is that the California current system will be changing. In particular, temperature across all the 3 models are going to increase. And there's going to be an intensification of upwelling and poleward movement of the center of upwelling in the future. Next. And so as part of the project, we want to assess how these changes are going to impact the California current ecosystem, and in particular, the forage assemblage and their dependent predators. Next. And we also want to assess how those impact not only affect the predators, but the fishing communities that depend on them. And we are focusing on the coastal pelagic species fleet, whose livelihood depends on this forage species. Next. And finally, we are going to assess if our current management system are robust to these changes. We have 2 project aims that reflect what I've been talking about. So the first is to assess the effects of climate change on the forage fish assemblage, the predators, and the fishing communities that are dependent on them. And then 2, we're going to develop a climate-informed ecosystem management strategy evaluation to assess the performance of current, but also alternative management strategy for the CPS species under a changing climate, shifting forage species composition, and varying predator populations. Next. The project team is here, so I'm the lead PI, but I work closely with my 2 co-PIs, Isaac Kaplan and Barb Muehling. It's a very interdisciplinary team, so we have members that are oceanographers, social scientists, ecosystem modelers, and fish biologists. And I'm not going to highlight everybody, but I want to highlight 4 important people, which are the 3 postdoc, Pierre-Yves Urban, Felipe Quesada Escalona, and Robert Wildermuth, and then Stefan Königstein. So they're supported 100% from this project, and they're doing a lot of the work, and so expect to see many papers from them in the coming years. Next. So here is our modeling approach. I've talked about the downscale projections of ocean conditions. So those fit into both species distribution models and a set of biological models. And so this ensemble of biological models range in complexity from the Atlantis whole ecosystem models that Stephanie mentioned at the beginning of the presentation to a model of intermediate complexity for ecosystem assessment, a MICE model, which integrates sardine and anchovy and specific predators, and then a single species sardine models. So all these 3 models make different assumptions and have different parameterization. So we are also using this ensemble of biological models as different operating models in our management strategy evaluation to capture some of that biological uncertainty, and the uncertainty specifically in the response of some of these forage species to climate. And then finally, output from this operating model is feeding into economic models that are being developed to assess the response of the CPS fleet to changing in forage availability, and as well to develop fishing community impacts. Next. So in relation to what the work that we've seen today, how do we see that fit in with the Future Seas project? We feel that trait-based approach are going to be extremely useful to refine functional groups and species interaction strength within our Atlantis ecosystem model. We also feel that the trait-based projection of future changes in prey-predator interaction and predator distribution will be extremely useful to improve the characterization of uncertainty in our model. So that could be a fourth ensemble member, and we would like to compare those projections with our current taxonomic approach. The work so far has proved invaluable to understand the drivers of variability in the Southwest Fisheries Science Center diet database. And so once we understand those past drivers, then we could infer future feeding links and develop scenarios for those prey-predator relationships for projections. The trait-based approach would also allow us to predict and understand the impact of invasive or emerging species into the California current system, particularly emerging predators, and their impact on our forage species of interest. And finally, in an ecosystem modeling context, it would again improve characterization of the diet of predators that are poorly sampled from our diet database. And the last opportunity would be to work with the LensFest project team to develop trait-based indicators of ecosystem change that might be relevant to management. Next. So that's my brief overview. Thank you for letting me be here, and I want to thank the entire project team from Future Seas. Back to Stephanie.
Thank you, Desiree, for that overview and context of how the pieces fit together. We want to say a big thank you to everyone that has attended the webinar today. Many of you have contributed to the project and also in discussions with us, and perhaps many of you are folks that we have not yet met or had the opportunity to interact with. So I'd like to extend the invitation and hope from all of us that spoke today that we would love to hear your feedback and engage with you about your thoughts on what we presented so far in terms of our findings and the trait basis of foraging interactions and how that information might tell us something about this changing Eastern North Pacific system that we are intently working on, or perhaps how it might be applied in other ways to issues that you are particularly working on. We'd invite you to contact us as well as to explore the data that we've made available open access online. And we'd welcome engagement with everyone on this webinar going forward, and also suggestions of opportunities for different spaces and groups that might also be interested in hearing about this work. And with that, I'd like to thank you again, and I'll turn it back over to Kayla.
Wonderful. Thank you all so much. This was a fantastic presentation. Thank you for walking through all of the results that you have so far. And I'm really excited to dive a little deeper into some of the trait database information that Miram was sharing. Also, I really love the analogy of going to the grocery store and trying to decide between an apple or a pretzel. But yeah, thank you all very much for the presentation. We have the contact information for all the speakers and research team included here on the slide in front of you. So please feel free to reach out to them or us at LenFest if you do have other questions or just want to get in touch with us. Additionally, next week on Thursday, December 8th, we'll be hosting a live Twitter Q&A with some of the research team. So if you log off after today and have some other questions, or if for some reason we don't get to all the questions that were entered into the Q&A today, you can ask those from 2 to 3 p.m. Eastern time and 11 to 12 p.m. Pacific time directly by tweeting directly to our Twitter account at LenFestOcean. Just use the hashtag at AskLenFest. So we'll have some of the research team join us again for that, and I'll include that information in a follow-up email that also includes the recording to this webinar as well. But for now, we can get to some of the questions that we have today. So first question is, were you able to account for any of the trawl sampling biases?
Maybe let Miram take that one if you're up for it.
Yeah, so I think for us, one bias that we know is kind of the biggest one is albacore actually are present in the system later when trawl sampling occurs. Trawl sampling is generally in June, May or June, maybe into July, and albacore don't start to show up until July. And our goal in this work was using the best data that we had available to us. And fortunately, in the California current, there are a lot of these trawl surveys. And despite that kind of difference in time, we kind of moved forward with that as we could. And but that is something that we absolutely acknowledge. There's that difference in time, and as well as the fact that the trawls don't necessarily sample everything that an albacore can sample. And through a combination of the community analysis work that Natasha did, plus some other work that we actually didn't present today for the sake of time, we show that the trawls are sampled quite differently. And in our interpretation of results, we do kind of couch some of this. There's some species they sample great, and we can provide outcomes on that. And other species that, possibly albacore might even be better samplers of them than the trawls are. So that's something we're, parsing out in the work.
Excellent, thank you. Next question.
I think Tasha's hand is up too. Did you want to add something?
Oh,
Tash,
I do see that now.
Yeah, I did, because I think this is interesting. This is obviously a really long discussion, and we are never going to get a boat at the exact same spot that a predator is eating food, and we'll never know the exact known truth of the forage community. But our colleagues at the Southwest Fisheries Science Centre, and especially Kat Nichols' analysis and soon to be published paper on albacore diet, does highlight that, at least for the environmental variables, the 6-month signal prior to when a sample was collected tend to be the most significant variables, rather than, for example, sea surface temperature from the same date and time. So 6 months prior to a sample was usually more indicative, which is why our group and our colleagues at Future Seas believe that we can get indicator information from these data sets.
Excellent, thanks. All right, next question. There's more and more science showing that the development of offshore wind will have ecosystem impacts in terms of zooplankton distribution, ocean mixing, and upwelling. To what degree is the planned developments of offshore wind energy off the West Coast incorporated into this analysis or study?
Yeah, thank you for the question. And I'd love to learn more about this particular area of development in ocean spaces. Our lens so far has been on characterizing communities of species in open ocean systems from this trait-based perspective. We haven't explicitly done anything with offshore wind development at this point, but our hope is that providing this sort of new lens on looking at the composition of what is already currently out there and what has been out there in the past, as we mentioned from the sampling data that we have access to in the system, that we might be able to look at changes in the trait sets of species that are there and how predators and ultimately their foraging might be impacted by shifts in those species. So we haven't explicitly considered that yet, but perhaps there could be multiple uses for this information as another layer to help explain changes in the system. And I look forward to investigating that link and would be grateful for anything else that you could share on that. I don't know if others want to add anything to that response.
No? All right. We also got their contact information, and they're more than happy to have a discussion about this. So we'll be sure to pass that along to you all. And then I realize that we're right at 3 o'clock, but we only have one more question left. So it would be great if we could get to that just real quickly. How did you decide which traits to include in your database and analyses?
I can start and then pass off. Thank you for the question, Sarah. This is something certainly our project team goes back and forth on. And there's the wish list of traits, and probably traits that we don't yet have hypotheses about. And then there's what is practicable to gather information on at the resolution that we'd like. And so we started with combing the literature and investigating hypotheses generated from observational studies, experiments, and just knowledge of the foraging behavior of albacore as a predator, and in general, foraging by predators in open ocean ecosystems. Generally, there are attributes of species that you can measure: their physical, the physical organism, their morphology, their behavior we can hope to observe or at least infer something about, as well as how valuable in terms of nutrition they might be. And so these are areas that there have been other studies in other aquatic systems and across ecosystems showing that there's some relationship between these properties and how predators are using prey. That goes back many decades before I was a scientist and draws on optimal foraging theory. And so after combing the literature and coming up with a set of potential morphological, behavioral, and nutritional characteristics that we'd like, we then see what we can gather data on and what's out there in literature, as well as what we could measure in the lab through photogrammetry, and what we could assess using modern techniques of looking at proximate composition. So we're always open to new suggestions of traits that might be valuable for providing insights on how predators are using prey resources. If that's something you're interested in discussing further, we'd love that. And also to ways of thinking about how we quantify those traits and measure them. I'll hand it over to Tash.
I think that was super comprehensive, but I wanted to add that technically it was also a working group effort with all of the staff and personnel on this project, but also with multiple discussions with Future Seas project scientists and biologists, and trying to figure out what were we interested in from a course perspective. For example, there were those habitat use traits, to see if there's a pelagic, mesopelagic, or demersal signal in their diets, and when. And there are traits that some people were less attached to than others. For example, I don't remember caring about transparency as a trait, but we can see from the data now that albacore tuna don't seem to eat transparent prey, which is interesting when there's a lot of work showing that we're going to be dealing with more gelatinous species and jellyfish under warming climates. So there's, they may continue to select for other prey. So there's a long list of traits, and they were developed as a working group. If you're interested, and as Steph said, we're always open to discussion on including more traits and also how to include those traits.
Excellent. Thank you very much. Anyone else want to add anything before we end the webinar? All good. All right, wonderful. Well, thank you again very much to our speakers, and thank you all for joining us today. Please follow up if you have any other questions, and if you're on Twitter, join us next week for our live Twitter Q&A with the research team. But until then, thank you everyone, and have a great day.